Macy Sherman.
Amazing. Macy, what lab are you with?
I am part of Dr. Milton's Comparative Animal and Marine Physiology Lab.
All right, and tell us a little bit about your research. So what is a take-home message you want everyone to know from ISTS?
Basically, diet plays a very big role in pollutant loads, specifically toxic elements within the body, and we're doing what we can to prevent that in the future.
Amazing. And what's one thing you're looking forward to doing while you're in Hawaii?
Cliff jumping.
Hi, we're here with
Gabby Carvajal.
All right, Gabby, what lab are you with?
I'm in Dr. Jeanette Weineken's lab.
And tell us a little bit about your poster. What's one take-home message?
It's really hard to sex hatchlings.
Agree. All right, what's one thing that you'd like to do while you're in Hawaii?
Scuba dive and whale watch.
Throw it up. Swimming right over to
Rachel Southerns.
All right, Rachel, what lab are you in?
I'm in Dr. Milton's lab.
Amazing. And tell us a little bit about your poster. What's one take-home message?
I think the biggest take-home message from my poster would be that plastics are a problem, that we need to do something about them if we want to save our sea turtle friends.
Absolutely, I agree. And what's one thing that you want to do in Hawaii?
Just enjoy it and really take it in and embrace the experience. Being
Love
here.
it. Awesome. Thank you. Hello. All right, we're here with
Ivana.
Hi, Ivana. What lab are you in?
I am in Jeanette Weineken's lab at the Marine Lab.
All right, and tell us a little bit about your poster. What's one take-home message you'd like the scientists to take home from ISTS?
I would say my one take-home message is that we traditionally think of sea turtle shells as these really strong protective structures, but we're starting to see through mechanics and some structural work that they're actually very flexible, dynamic structures that we're hoping to learn more and more about.
Very cool. And one more thing. What's one thing that you're looking forward to doing in Hawaii?
I am very excited to do the manta ray snorkel and just be one with the mantas.
We're here for the second day of ISTS research, and I'm here with
Dr. Samantha Kushke.
Hi, Sam. Whose lab are you in?
So I work with Dr. Tiffany Briggs, Dr. Mike McCoy, and Dr. Jeanette Weineken.
Excellent. And so give us a take-home message about the research poster that you are presenting at ISTS.
So the take-home is that this fungi that I study, called Fusarium, is indeed present on our beaches and has the ability to negatively impact not only sea turtle nests, but also sea turtles later in life, and could potentially impact, humans and plants and animals also on our beaches. So it's a pretty important fungi.
Yeah, I was gonna say very important research to have. And one more thing, what are you looking forward to doing while you're in Hawaii?
I'm supposed to go manta ray snorkeling tonight, so I'm super excited for that.
I'm going along. I'm here with
Kylie Harrison.
Hi, Kylie. Whose lab are you in?
I'm in the camp lab with Dr. Milton.
And tell us one fun fact or one important take-home message from your research.
I would say one take-home message is that plastics are in everything, even sea turtle eggs.
Reduce, reuse, and recycle. And what's one fun thing you're looking forward to doing while you're in Hawaii?
My fun thing is that I'm going on a 12-hour birding tour. Yeah, I'm so excited.
12 hours. All right, I'm here with
Heather Seaman,
Hi Heather, and whose lab are you in?
Dr. Sarah Milton's.
And tell us a little bit about your research. What's a take-home message that you'd like the scientists to learn about?
We all need to do our part in reducing our plastic consumption so it doesn't go into the environment.
Very, very important. And since we're in Hawaii, what's one thing that you'd like to do while you're in Hawaii?
The manta ray snorkel, of course.
Everyone's been saying that. It looks like we're going to have to go. All right, get ready for some science. I'm here with
Shelly Lenz,
All right, Shelly, whose lab are you in?
Dr. Sarah Milton.
And give a take-home message about your research you'd like everyone to know from ISTS.
Don't grow cells.
Oh, and why not?
I'm just kidding. They're really cool, but mine took 82 and a half days to reach their first passage. And that was just the first time I split them up.
And why are you growing cells?
To use for toxicology studies. So I utilize them to determine cytotoxicity ranges for different contaminants that are relevant to Florida's nesting leatherbacks. And then I looked at some values in the blood and used correlations to test biomarker expression in those cells as well.
Wow, very cool. And what's one thing you're looking forward to do while you're in Hawaii?
The manta ray thing seems fun. We're doing that tomorrow. So
Yeah, it seems very popular. I'm going tonight. All right, I'm here with
Sam Trail
and Sam, whose lab are you in?
and Dr. Jeanette Weineken's lab.
And give us one fun fact from your poster or one message you want the scientists to know from ISTS.
All right, so overall, loggerheads and leatherbacks have flippers that are shaped a little bit different, and that seems to impact the force that they produce over time.
So you would say different strokes for different folks?
That is the title, yes.
Amazing. All right, we're here with
Elizabeth Schulteis.
and Elizabeth. Whose lab are you in?
I'm in Dr. Jeanette Weineken's lab.
Very nice. And what's a take-home message from your poster, your research today?
Fungi can be detrimental to sea turtle mass.
Very important information. And give me one thing that you're interested in doing while you're in Hawaii.
I'm going manta ray snorkeling tonight, actually, so I'm very excited.
I'm going with her.
Yep.
Hello, welcome to ISTS. We're here with
Sarah Sexton,
and you are from what lab?
the Comparative Animal and Marine Physiology Lab, led by Dr. Sarah Milton.
Fantastic. So you're here presenting your poster. What would you say is the main takeaway you want people to get from this?
So I have found that phthalates, which are a type of compound that leach from plastics, may potentially disrupt the sexual differentiation process in turtle embryos, which could potentially lead to feminized populations.
A lot of ladies. Thank you. Are you looking forward to doing something in Hawaii?
Yes, actually tonight me and my lab are going on a manta ray snorkel, so I'm very excited.
I hope you see some. Thank you.
Hi, can you tell us your name?
My name is Emily Turla.
And whose lab are you in?
I'm in Dr. Jeanette Weineken's lab.
Can you tell us one cool thing about your research?
We found that 32 degrees Celsius was completely lethal to early embryos during development.
Boo! What's one thing you're excited to do while here in Hawaii?
I'm excited to go to Volcano National Park.
Welcome to ISTS. I'm here with,
Jeanette Weineken.
and what is your role at the Marine Lab?
I am the director of the FAU Marine Science Lab, and I work with the Glenn W. and Cornelia T. Bailey Marine Sea Scholars.
Our fearless leader. So Jeanette, can you tell us your main takeaway that you want people to know about your poster?
Well, the first thing is that sea turtles do not have a diaphragm like you and me. Instead, they breathe by actually bending their shell and then letting it relax. And when it relaxes, they exhale, and when they bend it, their lung drops down and they inhale.
Super cool. And aside from turtle stuff, is there anything fun you want to do when you're in Hawaii?
Oh yeah, there's lots of fun things, but seeing old friends and making new friends is always a good one. But I did take some time out to go drive around a little bit, and I really love the honey here in Hawaii, where the macadamia trees provide the nectar for the honeybees. So I went to Big Island Bees and got to see this place where I've been ordering honey for the last 8 or 9 years. And of course I bought more honey.
She got more honey. Awesome. Thank you.
You're welcome.
Hello, we're here at the last day of the poster session with,
Dr. Chelsea Venice.
and Chelsea, what lab are you in?
I'm in Dr. Jeanette Weineken's lab.
Fantastic. Can you tell us a little bit about your poster?
Yes, I'm going to give you a bite-sized summary. So greens and hawksbills off of Maui's coast, they eat the same red algae. They're able to do this and not compete for this resource because they have different face lengths that allows them to feed on this algae using different foraging behaviors. So multiple mechanisms of coexistence.
Super cool. And tell us, what's one thing you're looking forward to doing in Hawaii?
While I'm in Hawaii, I'm looking forward to spending time with my lab mates.